Okay, today I'm going to be talking about high throughput sequencing. So just a brief outline, talk a little bit about omics and the history of next generation sequencing, a little bit about the chemistry, which is important to understand, specifically focusing a bit more on Illumina than the other platforms, as Illumina is the type of data we'll primarily be using in this class. And then following up a little bit on file formats, we learned FASTA format last time, we'll be learning FASTQ today. So two acronyms to be aware of that you'll probably run into. NGS for next generation sequencing is by far the most common. The second one that I sometimes see is HTS, which is high throughput sequencing, although that one is definitely a little less common. So birth of the word genome. This is actually a surprisingly old word. I always assumed it was pretty recent because we've only been able to sequence whole genomes for a few decades. But it turns out it's actually from, they think the original first usage is 1920. Hans Winkler was a German botanist, and he proposed the expression genome for the haploid chromosome set, basically specifying the genetic material that's the foundation of any given species. So it is quite an old word from 1920. And now we hear genome and ohm being used all over the place, lots of omics. Biome is also an older word from 1916. And ohm in general, over time, has come to mean all of the constituent parts considered as a collective. So the genome is all of the genetic material considered, all together. This is completely unrelated to the word chromosome. The ohm and chromosome comes from a different word origin, which is kind of interesting. But in the 1990s is really when we saw this take off. Now we have transcriptomes, which is all of the transcripts, and metabolomes, which is all of the metabolites, and proteomics, which is all of the proteins. So that's, and now we have all sorts of other omics. It's actually getting a little crazy out there with people coining new omics terms. But in general, anytime somebody puts ohm on the end of a word, they're trying to say the full collective of the biological molecules that are in an organism or in a sample. So there have been, in general, 3 generations of sequencing that people kind of think about, 3 eras of sequencing. Originally, we just had sequencing. Nobody called it first generation because it just was sequencing, and that was primarily Sanger sequencing. There were gel-based methods in the 60s, but moving into the 70s is really the first time that we saw the Sanger method emerge, and that led to instrumentation and actually being able to do lots of reactions in parallel into the 80s and 90s. The chemistry is chain termination or dideoxy. We're not going to cover that in this class just because we aren't primarily working with that type of data. But if you're going to get Sanger data, which is still pretty common, we have a Sanger sequencing instrument on campus, it's worth kind of learning some more about how this works. Get 800 to 1,000 base pair reads, very high quality, over 99% quality. In 2003 is when we first saw this next generation of sequencing technology, sometimes also referred to as second generation. This was a lot more data, just a lot more data, and so much less expensive. Like prices just bottomed out per nucleotide. There were a couple of different chemistries. One is sequencing by synthesis, another one was called pyrosequencing, but all of these technologies were very limited by read length. Started out extremely short, 25, 35 nucleotides per read, looking at about 300 now, but still capped at quite a short read length. The third generation of sequencing, which is yet another completely different set of chemistries, came along around 2011. This is set apart, I would say, by being single molecule sequencing. They require no amplification, and they produce significantly longer reads, but have gone back to being more expensive per base. But in this case, like much more longer reads, so up to 90,000 bases in a single read. So that's a really different type of application that you can use with these types of long reads. So just some general information about how NGS works. DNA sample preparation involves the addition of defined sequences known as adapters to the ends of, sometimes the DNA is randomly fragmented. It can also be amplified or selected in some other way. So not necessarily randomly fragmented, but often. And this creates what we call a library. And that's just a very, very high level overview of a library. There's actually many, many steps to library creation. But basically you have a set of molecules, DNA or RNA molecules, and you have added adapters. And then the next step is immobilization. So the adapters are used to anchor the individual DNA fragments to a solid surface, like the last slide. And this is where amplification is required. So if amplification, it can be avoided in the library stage. It is often part of the library stage. But even if you avoid it there will have to be amplification on the slide itself. And in the case of Illumina, that's going to form these little spatially distinct and detectable sequence features that can actually be sequenced. And I'm going to give you a video to watch that's going to make this a lot more clear. And then finally, sequencing. So in the case of Illumina, your DNA polymerase synthesis with fluorescent nucleotides actually gives you that fluorescent signal so that you can take an image and based on the color of the fluorescence, determine what base is being added. So as this polymerase is adding bases, a complementary strand to your DNA molecule or RNA molecule of interest. RNA has been converted to cDNA. But anyway, so the polymerase is adding bases and they fluoresce in a different color, and that's how you can tell what your sequence is. And the cycle there with Illumina is the base is incorporated. As it fluoresces, there is an image taken that is washed, and then you go to the next round of incorporation, incorporate another base, image, wash, and you go through cycles to build up your sequence. I'm pretty terrible at explaining all of this, so I'm gonna, like I said, post some videos. But let's talk a little bit more about Illumina. I took a screenshot of their stock market share on Google today, and let me just tell you, I really wish that I had invested in Illumina about 10 years ago because they are have really just become a very dominant biotechnology company worth a lot of money. So almost $40 billion is their current capitalization right now. And part of this is because they have over 75% market share for DNA sequencing. They're really dominant. And why? Why are they so popular? They have a very, very low price, of course, is a big part of it. It's hard as researchers to say no to a good deal. Even with the low price, you get very high throughput. And they had another advantage that I think really set them apart from some of their early competitors, which is paired-end sequencing, which I'll explain in a minute. And here's a chart that just kind of shows, again, illustrates how influential next generation sequencing was. You can see starting in 2001, 2002, prices were coming down. Sequencing was getting less expensive. Advances were happening. But then as soon as Illumina enters the market, within just a few years, the prices just start plummeting. So really the amount of money you're spending for nucleotide is a mere pennies on the dollar of what it used to be. This is a really great figure from Lex Niederbrecht. And he's a great bioinformatician too. He has a blog and he has a Twitter account. And so if you like following people on social media, he's a pretty good one. And he posted this really neat figure of sequencing instruments and just showing, kind of plotting their read length versus how many gigabases you get per run. So this doesn't take into account price, but it's two very useful metrics. And I can move this. I think this is in everyone's way. So looking at this, I've highlighted some of the Illumina instruments up here. And this is why I think Illumina has really won over like some of these other instruments that you'll see down here. Those companies are no longer in existence, and those instruments are no longer on the market. And so what Illumina really did right was this very, very, very high throughput. And these lines kind of indicate how the companies have moved over time, or how the instruments have moved over time. And you can see Illumina has done a really good job of pushing their instruments toward many, many more gigabases per run. They have still been largely limited by read length and have not made a ton of progress on that, but they have been able to really, really increase that output, which has made them very, very dominant. Over here with the MinION and the PacBio, we're going to talk a little bit about those. These are these third generation technologies where we're seeing lower gigabases per run, but they have interesting applications because they have such very, very long rates. So comparing a little bit more within the Illumina platform, Illumina produces a whole suite of different instruments. If you're embarking on some sequencing, it's important to kind of have a feeling for what these might be and to talk to your sequencing core about which one fits your needs the best. The smallest sort of commercial level, there are actually really very small Illumina instruments that you can buy as like benchtop sequencers. But for a core facility or a company, the smallest you're generally looking at is a MiSeq. We're on the version 3 chemistry. The great thing about this instrument is that you can get reads up to 300 base pairs long. So quite a bit longer. That's the maximum length that has ever been offered on an Illumina instrument. So 300 bases is great. Here we see 13.2 gigabases per run. I got these prices off of the Duke Center for Genomic and Computational Biology. They have a nice little price estimator. You can play with tons of different parameters. So these prices, I tried to do like one library vanilla DNA sequencing. But it's really easy to change these prices with different, different types of runs. So take these with a grain of salt. You need to go price out your own experiment. Moving from there, the HiSeq 4000 is the HiSeqs have been on the market for quite some time. They've really been the workhorses of the Illumina platform for a very long time. Much, much, much higher throughput, so 100 gigabytes or gigabases per lane, but shorter reads. They max out at 150 bases, and indeed all of these higher throughput instruments are only doing up to 150 bases. So if you need the 300 base reads, for example, if you're doing metagenomics at 16S or ITS sequencing, you're going to be tied to a MiSeq because of that read length. If you're not tied down to that read length, you can move up to these higher throughput machines. As you can see, Illumina has, the HiSeq has 10 times as much data for only about 25% extra in price. So really getting a lot more information for the same number of dollars. The NextSeq and the NovaSeq are even newer and higher throughput instruments. The NextSeq has a slightly different chemistry that I'll mention in a minute that is worth doing a little bit of research if you decide to go with the NextSeq. It's a 2-die chemistry instead of a 4-die chemistry. Should be almost as high of quality, maybe a little less quality. In this case, the Duke website said 100 to 120 gigabases per run. I think you actually get more like 300, which is why this doubling in price is generally supported. For the NovaSeq, that is the newest, that is the biggest, that is the really crazy instrument. You're looking at more like $9,000 a run, but depending on how you set the run up, you can get just really, really, really vast quantities of data compared to the other instruments. So definitely be aware of all of that and talk to your sequencing core to pick the application and the instrument that is right. We do have a MiSeq instrument available at UTA that you can contract to put your libraries on and get sequencing done. There's also Veronica Brown is the woman who runs it. She will also do library prep for you as a fee for service. And she teaches a class that goes all the way through the molecular techniques from library preparation to sequencing, all the way to the bioinformatics and doing data processing. And I think it's focused primarily on 16S data. Highly recommend that class. I've heard really good things. I know it filled up this year. If you want to take it in the future, register early. So all of this is part of the Center for Environmental Biotechnology. They also have a bioinformatics resource center that I want to mention. Robert Murdoch is a bioinformatician over there, available for consulting. And he also has a lab with a number of computers that have some good software packages. I think they have 5 licenses of CLC Bio, which is a point-and-click software package, no command line. And that may be worth checking out, and it's a great resource. So some limitations of the Illumina platform. I've mentioned multiple times that shorter read length. With the MiSeq, you're tied to 300 bases. With the HiSeq, NextSeq, NovaSeq systems, you're tied to 150 bases. Additionally, Illumina has some really strong biases against sequencing through GC-rich regions or AT-rich regions. Those are usually underrepresented in samples or not sequenced at all. So if you're working on microbes with crazy GC percentages, something to be aware of. The error profile for Illumina is very, you're likely to see SNPs. So you don't see a lot of gaps. So you don't see insertions or deletions being introduced by the sequencer, but you do see a lot of SNPs, which is just bases being miscalled. Additionally, they are not evenly distributed. The errors tend to cluster at the ends of reads. So the beginning of the read and the middle of the read will usually be quite high quality, and then moving toward the end of the read, you'll start seeing an accumulation of errors. I will put this link also on the course website, but this is a video that I want you guys to watch and learn more about how sequencing works. They do a great job animating it, and I think it's probably a way better explanation than I could do justice to. And I also pointed you to a link if you're thinking about using the NextSeq. Since it's a 2-die instead of a 4-die chemistry, you can go read about that here. So one thing that's important for the read processing, for actually doing the data analysis for library construction is thinking about how sequencing works, especially the adapters. So this is an example from a TrueSeq library kit in which there's a universal adapter at the beginning. It has been ligated to your DNA fragment of interest. This can be from 150 to 500 or 600 bases. And then also an adapter at the end that has an index or a barcode, which I'll talk a little bit more about when we get to the part of the lecture about multiplexing. But the reason why this is important when you're going to process your data is that these adapters are artificial sequences that you are adding to your sample. You need to get rid of them. They are not from whatever organism or environmental sample that you're sequencing. So you need to know the sequence, and you need to know where they're ligated and how, so that when you get the data back, you can remove them. So 2 things to talk about with Illumina sequencing that are important to understand are paired-end sequencing and multiplexing. So let's start with paired-end sequencing. Illumina as a company, has long been aware of the limitations of their platform that have come about due to the shorter read lengths. And they've tried to find clever ways to combat this. And paired-end sequencing is the first one, and it's actually extremely useful. So in this case, we might have a DNA fragment here that is 500 or 600 bases, and you can sequence from the one end. This is your forward read, get up to 100 or 150 bases, and then you can flip the DNA sequence over on the slide and sequence it from the reverse end. So you actually get 2 reads from the same DNA fragment, one starting at the beginning and one starting at the other end. So why would this be helpful? Well, knowing that those 2 reads, how they're oriented against each other, is actually a lot of information. So take, for example, this alignment to a reference sequence over on the right. Imagine we have a region of DNA that is repeated almost exactly in 2 locations. If you only have a short read and it originates entirely from that repeat region, you don't know which one it came from. I say there's a mutation in it. You don't know if it's the upstream repeat or the downstream repeat that actually contains the mutation. However, if you have a paired end, the other end from the other ends of your DNA fragment, if that maps to a unique region of the genome, that anchors your location. And now you can figure out, based on the relationship of those 2 fragments and how far apart they are, which of your reads originated in the upstream repeat and which originated from the downstream repeat. This turns out to be really useful for not just reference mapping, but also for assembly and all sorts of bioinformatic applications. There is another type of sequencing that you may run into called mate pair sequencing. This is much less common. It's very expensive. It's a pretty finicky molecular protocol, but it can be useful. So people do it. This is where you create a library that has much longer jump distances. So instead of starting with a fragment that is 400 or 500 bases, you start with a fragment that is anywhere from 2 kb to 40 kb. And then you, the way the library gets constructed is significantly different. And instead of the reads being pointing in from the ends of the fragment inward, they're actually from the edges of the fragment back out to the ends. So it's just important if you get mate pair data to realize that the reads are pointing the opposite direction. So they're pointing out. The reads are going from in to out instead of from out to in. So Usually if you have mate pairs, you're going to be using software meant to process mate pairs, and the software is going to know to handle that. Just in case you run into mate pair libraries, that's what's going on with those. Very, very long distances between your fragments. These are great for assembly. The only time I usually ever see these are for genome assemblies, and they're kind of going out of fashion as we get better ways to do really complex genome assemblies. So I talked a little bit about this already. Paired ends and mate pairs can be used to sort out the number of repeats or the sequence of repeats in genomes. Basically because one end, hopefully if one end is in a unique region and one end is in a repetitive region, that gives you an anchor point to figure out how many repeats you have and where they are in relative location to your unique regions. Okay, so I also want to talk about multiplexing. This multiplexing is great because we talked about how some of these instruments can give you just tons and tons and tons of data, many, many gigabases. And maybe you have 60 samples and you don't want to get 300 million reads from a single sample. You want to split those reads across all of your samples. Well, the way we do that is through multiplexing. And it's basically you're going to use your library preparation to allow you to downstream sort out what reads came from what libraries. Okay, so let's go through this. Let's say you have a pink sample and a green sample. For your pink sample, you're going to ligate on the standard universal barcode for Illumina, but with it, it's going to come with 6 bases, in this case, CGA, TGT, that are attached to that adapter. And every single read that came from your pink sample is going to have that standard adapter and then CGA, TGT. On the green sample, you're going to ligate on an adapter with a different barcode. This one's going to have the same original starting point so that it will attach to the glass slide appropriately. But at the end, you're going to have a different sequence, so TGA, CCA. And that's going to be in between your universal adapter and your actual DNA fragment that came from your sample. So you can do this for not just 2, 3, 4, 5, 10, 100 samples. They each get their own unique barcode. And once the libraries are made, you can mix them all together and put them all in the sequencer. The sequencer isn't going to know the difference. The sequencer is just going to sequence whatever is loaded into it. But then on the back end, when you get all the data back, the sequencer will have read through those barcodes. So you use software and you take and you look, you say, if right after the adapter, I see a CGA TGT, that came from my pink sample, I'm going to put it in a separate file that says reads from my pink sample. If instead you see a TGA CCA, you say, oh, that's a barcode from my green sample. I know that these reads must have originated from DNA from my green sample. I'm going to put those in a separate file. This is called demultiplexing. And most of the time, the sequencing facility is going to do it for you. Sometimes they aren't, and there are command line scripts to help you do this. There are barcode kits with up to 384 barcodes. I've heard of people doing even more than this. I don't know how common that is. I think at UT they have the 96 barcode kind of standard barcode set. It can be really beneficial to select your barcodes wisely. You want an even distribution of nucleotides per cycle. So, and so each base in Illumina is a cycle. So if the very first barcode, if every single barcode starts with an A, that causes problems on the instrument. What you really want is kind of a roughly equivalent number of A's, C's, T's, and G's. So at each position, the first position of the barcode, the second position of the barcode, the third position, you want to make sure the sequences are very diverse. So you wouldn't want a barcode that's like A, and another one's like A, G. Those are bad barcodes. You want them to be complex and to differ at lots of locations within the barcode. Okay, so that's everything I'm going to say about Illumina for the time being. Just to touch on some of these other technologies that you may run into. PacBio is probably the most popular third-generation sequencing platform. It was commercially released in 2011. And their libraries are called SMART libraries or SMART cells. You'll see, and that SMRT is single molecule real time. So that's really what their claim to fame is. The sequencing is not done on an amplified molecule, that's actually a single molecule that's being sequenced, and it's happening in real time. So you're getting the data in real time. It's much more expensive per base than Illumina, but the trade-off is you get very, very long reads, which has some nice applications. In addition to very long reads, however, you will be dealing with a very high percent error. I think it's about 15% error still. So 1 out of 6, 7 bases is wrong. However, the error is truly random. It is not supposed to be more pervasive at the ends of reads. So having truly random error makes it a little easier to deal with statistically. But it's also a slightly different type of error. You're more likely to see insertions and deletions than you are to see SNPs. So again, just very, very different, kind of the opposite of the Illumina platform. Very little GC bias. And another cool thing is, because of the way the chemistry works, you get the methylation status of each DNA nucleotide for free. So you don't have to do anything different in your library prep. You just do PacBio sequencing and you get whether the nucleotide is A, C, T, or G, and you also get the information about whether or not that nucleotide is methylated, which, if that's something you're interested in studying, is nice. Also worth noting, it requires a large amount of high quality DNA. So if you're working with degraded samples or very low quantity RNA samples or DNA samples, this can be not the best platform for you. So here is a distribution of read lengths. So in Illumina, you always get every read is exactly the same length. If you have an Illumina library of 150 base pair reads, you are going to get 150 base pairs for every single sequence. PacBio is different. Sometimes the sequences are quite short, and sometimes they're very, very, very long. So this is the distribution. So you can see there's quite a few. The bulk of your sequencing is probably going to be below 20,000 in each sequence in length, but you do have quite a bit of the library in this kind of a 30 to 50 KB range. And then your top 5% of reads are very, very, very long, and you will occasionally even get reads up to 70, 80, 90 KB, which is, you know, a lot of different applications. Another third generation sequencing company, Oxford Nanopore, has become quite popular. The Min Ion is one of their flagship products. It's a sequencer so small you can hold it in your hand. And I got to see someone demo this at a course a few years ago. It's really neat. They're much of the sequencing and the library prep relies on disposable parts. There's no fluorescence and there's no polymerase, so they're able to scale way down. And the way they basically are looking at how ion current disruptions, so as a DNA is feeding through a nanopore, it disrupts the electrical current of the membrane that the nanopore is embedded in, and they can measure that. It's kind of a cool technology. I'll post a video. It'll make way more sense once you watch the video. No fixed runtime. So one of their marketing things is that, oh, you just run it until you have enough data to answer your question. Just let it go. And then once you've answered your question, turn it off. It's long reads. It's not as long as PacBio. I think they're in the hundreds to thousands of bases, but maybe not in the tens of thousands of bases. And I'm going to move this over again so you can see. I was just on the website and I wanted to put a picture of this. This is their new Smidge Ion coming soon, and it just looks really cool because it's just plug it straight into your phone and do some sequencing on the go. So they're really trying to lead the market. This company is very devoted to leading the market in sequencing in a clinical setting, or sequencing in the field, or somewhere where you're going to go out and you're going to collect data on the fly, and it's going to be really convenient. So definitely something to keep your eye on. Very exciting stuff. Yep, I will post a video that basically shows how the DNA is like ratcheted through these molecular nanopores, and as it goes through the pore, it creates a current, and the current is measured, and that's how you get the ACs, Ts, and Gs detected. So let's talk a little bit. Let's get from the raw data to the ACs, Ts, and Gs that actually arrive from the core facility. There's a step in between. So if you're working with Illumina or PacBio, it's actually image data, because those are fluorescing nucleotides. So there's a camera, a very high fidelity camera, capturing those minuscule fluorescent dots. So those images have to be converted to ACs, Ts, and Gs. So it has to be converted to text. This is something you will probably never have to deal with. The vendor of the instrument supplies the software. It usually comes with the instrument. There's a computer attached to the instrument, and the computer does this. The image files are not usually kept because they're giant. They eat up too much room. So once the ACs, Ts, and Gs are called, those raw data files are usually gone. But one of the reasons to talk about this is because as the software is taking these images, or in the case of a MinION, the little current signals, and converting it to bases, to text, it can do some analysis and say, you know, the signal is really strong and clear. I think this is very high quality base, and it can assign it a high quality score. Whereas if the signal gets a little fuzzy, or it looks like something has gone wrong, or the image isn't clear, it isn't bright enough, the software will actually assign it kind of a low quality value. Say, I'm not actually sure if this is an A. I'm going to give it a 1 out of 10. It's probably an A. Or, you know, this is definitely an A. Super clear. Picture looks perfect. I'm going to give it like a 1 out of a million. So this is how we can actually get quality scores assigned to each and every base that comes off of the sequencer. So why would we want to go back and learn all this stuff about how sequencing works? Especially, I mean, I don't even have a bench for my lab. We only do bioinformatics. So why do I care about this? Because doing bioinformatics actually involves you understanding the data and how it's produced to do a good job. You need to understand what biases and what errors are in your data. You need to spot nonsense. Sometimes sequencing facilities mess up and send you the wrong reads, or sometimes instruments have problems and you get really bizarre results. If you don't understand how the instruments work and how the libraries are prepared, you may think that's indicative of something biologically real when it's not. So you really need to be able to dig in and make sense of your data. Also, if you're planning for robust statistical design and good experimental design, you need to know how many reads am I going to get? What kind of depth of coverage am I going to get? Am I designing an experiment that is powerful enough and generating the right kind of data to answer my question? And that involves understanding the chemistry and understanding the instruments. So I think I already said this, but I'll say it again. Always get the sequences for your adapters and your barcodes for later analysis. In general, Illumina has standard adapters. There's kind of this public letter where you can go look at those adapters and pull them out and use them to trim them out of your data. But any kit you use for library prep should come with those sequences. Okay, so previously in the last class period, we learned about the FASTA format, or FASTA format. And today we're gonna learn about the FASTQ format. Q is for quality. So last time, there was a header and there was a sequence, and then a header and a sequence, and there was no quality information. It was just, this is what it is. For FASTQ, which is the format that you'll get for Illumina reads, there's a lot of information about that quality. How well was the base called? So this is what it looks like. Upon first glance, quite intimidating. It looks like a whole load of nonsense. But if you understand the structure, we can start parsing this apart and actually understand what it means. So the first thing to know, in a FASTQ format file, a read is always going to take up exactly 4 lines. Previously in the FASTA format, reads could break, they could be over multiple lines. That is not happening here. In this format, it is always exactly 4 lines. If you get a FASTQ file, and we learn this command, we learn WC-L, that is the line count command. If you do a line count on a FASTQ file and the number you get back is not divisible by 4, your file has problems. There are exactly 4, no more and no less per read. And so let's dig in and look at one of these. First of all, the first line, the first symbol is always an @ sign. So let's take this down to just a single read. First line, first symbol is an @ symbol. So in the FASTA file, it was a greater than. in a FASTQ file is an @ symbol. Why did they change? I don't know. They don't ask me these things. But then the rest of the line is very similar to a comment or an identifier line in FASTA format. Unique sequence identifier. The first thing on the line, all the way up to the very first white space, has to be a unique sequence identifier. After that, you have optional description. In the case of Illumina, it actually usually has the instrument number and the location of the image on the slide where it was taken. So for troubleshooting at the core facility, there's some good information in there, probably not super useful to you. There may be other information. If it's a paired in library, so it's a read one and a read two, or a forward and a reverse read, usually that information is going to be embedded in there somewhere. You may see the length. All sorts of different things can appear on this line. That's not important. The important part is the @ symbol and the unique identifier. Moving down to the next line, that is the sequence. Straightforward, A, C's, T's, and G's. If there's an unknown base, it will be listed as an N for unknown. But in general, the only thing that should appear on this sequence is, on this line, is the sequence. The next line is a totally useless line that none of us know why it exists. Again, they don't ask me to make these things up. It always begins with a plus sign. That is the only rule for this line. Sometimes it is a plus sign with nothing else after it. Sometimes it is a plus sign and they repeat all the information from the previous comment line. So in this case, and this is common for Illumina, it'll either be a plus sign and nothing else, or it'll be a plus sign and then the unique identifier and all the rest of the stuff that was on the first line. This eats up a lot of space and kind of drives me crazy. I don't know why it's there. And the fourth line are the quality values for each base. So this is a one-to-one correspondence. This first nucleotide C has a quality value of B. The second nucleotide A has a quality value of A. The third G has a quality value of C. So they correspond. So these quality values are basically, they're like a secret code. They consist of numbers, symbols, and letters. And you kind of have to decode them to figure out what they mean. So let's get our decoder rings out and figure that out. So quality values, going back, way back to the 1990s, there was a piece of software called FRED, P-H-R-E-D, that was used to call bases from Sanger sequencing machines. And this quality scoring system kind of became the standard, and it is to this day still the standard in how we think about qualities of bases. It's calculated completely differently, but the numbers mean the same thing. So FRED quality scores are logarithmically linked to error probabilities. So in this case, a FRED quality score of 10 is a 1 in 10 probability of an incorrect base call, so about a 90% accuracy. Moving up to a FRED quality score of 20, that is 1 in 100, so a 99% accuracy. I think back in the Sanger days, you go all the way up to 60, and that would be a 1 in a million probability of an incorrect base call, so 99.9999% accuracy. Yeah, but these are numbers, and that's not what we saw on the previous slide. So how do we get from the numbers to these codes, these codes that are in the FASTQ file? Well, first, let me tell you why. Why did we change? Why did we not just use the numbers? And that's basically because storing all of those quality values was just taking up way too much space. And this is an example where if we have A, C, T, G, A, T, C, if you wanted to score an individual numeric quality value for each of those bases, you have to have 10 space, 15 space, 25 space. So you've gone from 7 nucleotides. You now need 21 characters to store their quality values. So it's just a very bloated way to store quality values in a file. It was just taking up a lot of space. So instead they said, well, let's assign the numbers to symbols. We'll create this little code. And then your quality value could just be JOXOLB. And that'll be, we'll know that the J means 10 and O means 15 and X means 25, but it'll be much shorter and it'll make our FASTQ files much smaller. So that was the general idea about why should we develop this new code. Unfortunately, it wasn't super well implemented. It went through some problems. This has been sorted out for about 6 years, but what happened initially is that Illumina, so before Illumina was Illumina, it was a company called Celexa. And they came on the scene, and they have this next generation sequencing. They reassigned all of the Fred quality values to numbers and letters and symbols. Okay, great. They then updated their chemistry a few years later, and they reassigned new numbers and letters and symbols to different quality values. And then they did it again, and then they did it again. And then everybody in the sequencing world was just completely fed up. And so starting back in, I think it was about 2010, 2011, we went back to the old Fred base, the original assignment of symbols, letters, and numbers back to the original Fred scores. And so there's this very complicated chart on Wikipedia that you can go look at. In general, you're not going to need to worry about it unless you're working with data that's like 6 to 8 years old, in which case I would check and try to see if you have any problems. But for our purposes, the only line you care about is this red line. So this is the current standing, and this is everybody has standardized across the board now. We're starting from the exclamation point up to the letter J. Those correspond from 0 to 41. So if you saw, for example, an I, you would know, or no, yeah, I think it ends at I. If you saw an I, that would be a Fred score of 41. If you saw an exclamation point, that would be a Fred score of 0. And looking at these in order, you can go, oh, Fred score 1, 2, 3, 4, 5, 6. So the shorthand here is that if you open your FASTQ file and on the quality line, you see lots of uppercase letters, those are corresponding to Fred scores in the 30s, which are very good. So 99.999% accurate. If you see lots of quality scores that are not capital letters, you know you're dealing with significantly lower quality. So down here in this range. And so that's kind of a shorthand. Like I said, most of you should not have to deal with any of the old Illumina quality scoring because it was kind of a mess. But in case you do have old data, that is something to look into. All right, so that is the overview of high throughput sequencing. We're gonna see adapter trimming and quality values a lot more coming up. If you guys have any questions, feel free to email me, or we can also do questions next time we're in class. All right, thank you.